Thank you for joining me today in my presentation on Sanger sequencing and analysis. I'll first start off by showing you our DNASTAR Navigator, which is a portal to all of our applications. As you can see, we support many workflows and various applications in our suite, some of which include assembly and analysis for our next generation sequencing data, and as well as for Sanger assembly and analysis. And with that, we will open up SeqMan Pro. Notice that if you hover over any one of these modules, you'll get a summary of what each module does. And here we can see that SeqMan Pro is the right tool for assembling our data for Sanger sequencing and analyzing next generation alignments and assemblies. So with that, I will go ahead and open the SeqMan Pro module here. And when this program first opens, you will see two windows. One is the config window, which I'm pointing to right now. And this window will contain a list of all of your contigs and additional information about them, such as their length, sequences, and positions. And the bottom half of the window will contain all of the sequences that went into those specific config or contigs. The main window here on the left is for the unassembled sequences window, and this is where you will import all of your data, your reference sequences, as well as your raw reads that you would like to align or assemble. And then within this window, we have additional options, additional buttons right here. These will allow you to assemble your data or choose a type of assembly you would wish to do. It has shortcut commands to adding your data, as well as change some options for trimming and marking reference sequences as reference. And on the top, we have the file structure. You have the file menu and additional options for advanced features I'll walk you through in the software. And with that, I would like to also mention there are multiple ways you can import your data into SeqMan Pro. If you have data downloaded or stored locally onto your machine, you can simply click on Add Sequences or go to File, Sequence Add, or you could download it directly from NetSearch, where it will connect to the NCBI database through the text search option. And if you open this, you can pick from different databases from this pull-down menu, or you can, right here is the different databases. And from the pull-down menu here, you can choose which field to search. By default, it's selected as all terms, and then you can type in either the session number or a keyword, and then you can click on search, and you will see a list of sequences displayed, which you can then import directly into SeqMan Pro. I have some local data on my computer, so I will go ahead and cancel out of this window. And now I will click on the Add Sequences option. And first off, I will start off with a reference-guided alignment of influenza virus. And here are my reference sequences for all the different segments. And I will just highlight them all and choose Add, and then Done. And now I will go back into Add Sequences, go up one folder, and choose my raw reads, my Sanger reads. And I'll import the two FASTA files, and then click on Done. Notice that on the top I have my reference sequences, and on the bottom I have all the reads. And next to each read, you will see its limits, sequence length, and you can see if they have quality scores or not in the other columns. Now, before assembling, aligning your data, you will have to make some adjustments to set up the project correctly. First off, we will need to mark our reference sequences as reference, and to do that, you would simply highlight all of your reference sequences if you have multiple references, and then click on this mark ref, and this will italicize your reference sequences, so it's easy to point out where your references are in the sequences view in the contig window. And the next step would be to do a vector scan on your data. So if you know your reads have vector contaminants, you can highlight them all first, and then you can pick your vector from this list. And I will pick Janus here. And by default, you'll see a question mark next to each selection. But if you go into options and do a scan all, you can see the scanning options that it will use. It will trim sequence ends and scan for vector. And if it finds that particular vector in your sequence, you will see a check mark next to the vector name, as I'm pointing to right now in this particular sequence. And if that vector is not found in your data, you'll still see a question mark. And once you've scanned for vector, you can go into the project menu and see a trim report. And this will give you the details for what was trimmed and the quality and the trim length, as well as the post-trim length. And you can scroll down and get a summary for each individual read. And then you can get an average on the bottom of the window. And we're almost ready to run the alignment, but before we do that, we can go into parameters to make sure some of the options that we want for our assembly are correct and are applicable to our data set. So to do that, you would go into the project menu and then into parameters. And from here, you will see a list of options that you can choose from for optimization of your alignment or assembly. The most important option is the assembling option on the left. And here you can specify the match size, the minimum match percentage, and the gap penalties. You want to make sure that you have the Pro assembler selected, which is a more robust assembler. It uses a more modern algorithm, whereas the classic assembler is for reproducing older projects from many, many years ago. Pro assembler is the best aligner we have right now in SeqMan Pro for Sanger data. And if you're not sure that you need to change your parameters, or if you would like to change your parameters but not sure how to improve your assembly, you can always click on this question mark on the bottom of the window, and that will take you to the help menu, and it will explain what each parameter does. I'll close this window. And if at any time you have questions regarding specific parameters or are not sure, you can always contact us directly, and we can help you with those as well. And you can go down the list and change the consensus calling if needed. You can specify paired end reads under pair specifiers. By default, I have it turned off. And you can also change other options in Snip discovery, some primer walking parameters, and others. I will leave the default settings as is, since these are tested to be the most accurate parameters for most alignments and assemblies. I'll go ahead and click on OK. And to begin the assembly, you would simply click on Assemble on the top left of the Unassembled Sequences window. and it's just going through the contigs. And now, the first thing you'll notice is this report window. This has a summary of the alignment. It will give you the location of where each read was placed and the contig it was placed in. And you can go to the top or scroll down, and you can look at specific reads for the contig you might be interested in, depending on the workflow that you're doing. And then you'll get a total assembly time. We can close this window. You can always go back to this window from the project menu and then report. We have additional reports. If you go into statistics, you'll get a summary for each contig, its total length, with and without gaps, and other information. I'll close this now. And this is our contig window. This is where our list of contigs are and all the sequences. As you can see, we have all 7 segments listed here. And the sequences, both reference and data that went into the contigs, are listed on the bottom half of the window. You can change the name of each contig depending on the reference name. And to do that, you can simply click and hold and then change the name from the default contig 5 to anything you like. You can name it, you can have it the name of the segment for the reference, or a chromosome name, or customize it to a gene if it's a gene, or anything else that you prefer. I will leave it as default for now. And I will also want to mention that all of these columns are sortable. You can sort based on name by clicking on it, by length, number of sequences, positions, and other information. And you can also move these around. Here. And customize the window to your liking. We will focus on contig 13 now, and to open this contig, you would simply double-click on it, and this will open our alignment view. And the alignment view is where you will look at all of your read data, look for contigs, conflicts or SNPs, and you can browse through each chromosome or segment in this case for the alignment. On the top, you'll get your current position where your cursor is blinking. If you move this around, you can see that's changing, and you will get the depth at that specific location. On the top right, you'll get the total length of the contig you're working in, and then we have the coordinates ruler. And under it, we'll have these triangles, which you can expand for translation or consensus features. On the left, we have zoom in and zoom out tools and some toggling tools that will allow you to summarize and hide names for larger alignments. And I'll go through those as I walk you through this window. So if you wanted to view the translations for your consensus, you can simply click on Translate, and you can see all 6 reading frames for top and bottom strand. And if you click on the triangle next to consensus, you can see the trace majority. And for the features, you will click on the triangle next to the reference name. As you can see, this is italicized. Note that by default, the reference sequence is the consensus sequence. And if you wanted to generate a true consensus, which is a very commonly asked question, you would simply need to unmark your reference sequence from the contig window. And to do that, you can switch back to the contig window and right-click on your reference and choose Unmark Reference Sequence. As you can see, this will generate a true consensus, and the read, which was previously used as your reference, will be taken into account for the newly generated consensus. I will switch this back to the reference sequence. Actually, there are a couple sequences here that are unmarked. Okay. And there's another view I wanted to show you, and that is the strategy view for the coverage report. And to do that, you can simply right-click on your config and go into strategy view. And the window is designed similarly to the alignment view, where you have some of the zoom in and zoom out tools and toggling tools on your left. And on the top, you have the ruler, you have the coverage threshold, the depth of coverage, and as well as a list of features. And on the bottom, you have the read summary and the directions they're going in. And please note that both of these windows are interactive with each other. Any position you go into one window, it will jump to that spot in all views that you're in. So this way you can easily focus on areas of less coverage or more coverage, and so on. Now we'll go ahead and hide some of the translations here. And one other window I wanted to show you was the feature list, where you can get a list of all the features. And to do that, you simply highlight your specific config that you're working on and go into features and then say show feature table. And this will show you a list of all your annotations. This example only has 3 annotations, but it's also interactive with your other windows. So if you click on one feature, it will highlight that region in both the alignment view as well as the strategy view. Okay. And I'll go ahead and close this. And now I wanted to mention that by default, when you see these red bases, those are indicating conflicts with the consensus. And you can quickly get an overview of all the conflicts that occur while scrolling through. Or you can go into the edit menu and choose find conflict. And this way you can go choose to find a conflict in either the consensus or all sequences in the config or the selected sequence. We will leave it at consensus and say find next. And here we have found a conflict. And on the bottom, you'll see a summary of what's found. If you want to find the next conflict, you can toggle between these arrows, and it will just search for the last criteria that was specified in the edit find menu. You can also perform other searches, such as you can jump to a specific position by going into go to position from the edit menu. And here, if you type in any position, it will jump right to that location. 50, for example, we're right at 50. One thing I wanted to mention about the consensus sequence and the trace majority. By default, the consensus will take the reads that you have or the bases you have at a specific position, and it will apply calculation to generate the best consensus call for that region. And this is especially true when there are cases of heterozygosity in your reads. For example, the actual call was in R, but R somewhere called NA based on some of the trace data. And I'll go into that in a later assembly. I'll show you the trace data and show how that decision is made. Now I will jump into our Snip report to do variation analysis. And to do that, you'll simply go into Snip menu and choose Snip report. And here you can see a quick summary of all the SNPs that are found in this project in this config, and you can switch between the two options on top, between all found SNPs and SNP summary. The all found SNPs will give you a list of all the SNPs and their positional information and additional details in a quick snapshot. And the SNP summary will include some of the more filtering tools so that you can focus on your specific SNPs of interest. Again, all of these columns are sortable, and you can also move them around to customize them. You can also use some of the hidden shortcut commands. If you right-click on the gray area here, you can sort based on specific criteria, and you can see a list of all of these columns listed here as well. And you can also choose to hide columns that are not of great interest to you. For example, if you choose to hide some of the counts, that will get rid of those specific columns. And this way you can focus on only the information you're interested in. Also note that we have a summary of all the SNPs found in this config, and this number will update as you make any changes to the filtering. So if you wanted to change the SNP percent filter, minimum of from 0 to 25, you will notice this number has updated as you're making that change. And you can also add additional criteria such as a minimum depth of 10, for example. And this will further narrow down your report to 50 SNPs. And then you can add additional criteria such as the maximum distance from a coding feature. Let's choose 100 from here. And it looks like these are all within that range, so this did not update. Now we'll cover some of the columns. We have the reference position, you have the config, you can differentiate between a SNP and an indel. You have the called base, the percentage, and then you'll also see the codon, if there's a codon change, feature, and you can have the depth of coverage, and you can go down this list to look at all those changes. Note that this window is also interactive with the alignment view and the strategy view. Most users like to have the SNP report side by side with the alignment view, and the benefit of that is you can quickly scroll down the SNP report and look at the conflicts. And as you're going down this report, you can either confirm or reject the SNP. The question mark by default next to each SNP means it's a putative SNP. If you click on this once, it will confirm it, and it will auto-annotate your reference sequence. So as you're going down the list, you're rejecting and confirming SNPs, you're building your reference sequence for maybe a future assembly. And you can keep going down the list and do this for all other SNPs.
Farhan, we have a few questions that we'd like to get to now. It looked like this project uses FASTA data. Is it possible to load ABI files into SeqMan Pro?
Yes, you can definitely load in ABI files. With the ABI files, you will be able to see the trace data in your alignment view. And the next project that I will go into will be working with ABI data, and I'll be able to show you some of those features. For this project, you're correct, they are FASTA files.
And then another question about the vectors that you talked about toward the beginning. There was a list of vectors that you could select. Is it possible to add your own vector data to SeqMan or Lasergene?
Yes, definitely. The list of vectors we have listed are the most commonly used ones. However, if you have any custom vectors, or if there's a vector in there that's not listed, you can definitely add that to SeqMan Pro, and it will use it for trimming out your data for your reads. And we have steps in our help menu, and we can guide you through that process as well. It's very easy to do.
Great. And then one last question before you get back into the demo. The SNP table that you're showing here, is it possible to save this or export it to another program?
Definitely, you can save this. This will save as a text file from the File menu. So while you have this open, you can go into File, save the report for the specific config. If you wanted to save all SNPs for all configs, you can run that report as well. You would simply click on this unlocated configs and then run a SNP report. And then here you can see we have the full summary of all configs, and we have a list of all the config names. So yes, you can definitely do that.
Great, thank you.
And before we move into our other workflow for de novo assembly, I wanted to point out that we also have BLAST capability. So if you wanted to identify a specific config, you can click on that config, and you can go into Net Search and then BLAST selection. You can then choose the type of BLAST you wish to perform from the pull-down menu, and you can also then select the database. We will stick with the default NR database for this. Click on OK, and this will connect to NCBI and list all the hits that it collects, and I'll go over that as soon as this populates. And depending on the time of the day you do and the connection speed, it could take a few seconds to a few minutes here. Hopefully this will be quick.
While we're waiting, another question that came in, I think you may have mentioned this at the beginning, but can you just sort of explain, is it possible to use next-gen data with SeqMan Pro?
Definitely, you can perform all of your analysis of next-gen data in SeqMan Pro, as with Sanger data. So definitely that is possible. We have tools that will allow you to generate project files for next-generation sequencing assembly and alignments. And your analysis will be performed in this program, SeqMan Pro. So Sanger sequence is just one of the many workflows that we support in SeqMan Pro. So you can definitely work with all major next-gen sequencing data technologies, such as Illumina, 454, and iTorrent, and PacBio. So now our BLAST results are up, and I'll spend a minute here covering this report. On the top, you'll see columns for support, the score, the expected, and you have the query, and the left and the right, as well as the matches. You can scroll to the right and look at the description of your hit. On the bottom, you'll get a summary of the identity match. Here, this is our query, and our hit is listed right under it, and you can see where the matches occur. And for this, it has identified this is segment 2, which is correct. If we go into our config 12, we can see that this is definitely segment 2. So you can do that for any part of the actual chromosome as well. If you highlight a specific region within this config, you can blast that particular selection as well. It doesn't necessarily have to be the entire config. So with that, I will go ahead and close this window, and we will move into our next workflow for de novo assemblies. And to start off a new project, you would simply go to File, New, and similarly to before, to add your data, you would click on Add Sequences, or you can import your data from the new text search. So I'll go ahead and click on Add Sequences. And I'll load in some of my paired end reads. Highlight them all, click on Add, and then Done. And I will apply a vector scan to these. I'll choose Janus from the list. And before I even do the trimming, I will go into our project and parameters. And I will just want to ensure that all of the parameters I want are correct for the assembly that I'll be doing. Now I mentioned I loaded in some paired end data. By default, the paired end is turned off, so you would want to go into the pair specifier option and make sure to select one of these patterns to compensate for your count for your paired end reads. And I've selected the default first one. And if you know your paired end size, by default it's set at 0 to 3000. If yours is more, you can always change this to more or less. I'll stick with the default parameters. And I'll make sure that the Pro assembler and all these parameters are also correct. And then I will click on OK. And now I will go ahead and do the vector scan. And note that if you don't do the vector scan before the assembly, it will happen automatically during the assembly. So if you forget, that's okay. It will still happen. And I can go into Options and do Scan All. And one thing I did not mention previously is that if you double-click on any one of these reads, you can see your chromatogram as well as the area that was trimmed. If you did not have a chromatogram, then you would just see your sequence and the trimmed area. And here we can see our bases, the chromatograms, and where the trimming occurred. This vector arrow indicates the position where it trimmed to. If you wanted to untrim this, you can click and drag this to the left, and it will undo the trimming, but we'll move it back. And you can scroll through the entire read like this. And here's our other 3' end of the trim. And you can zoom in, or you can zoom out. You can also view the quality scores here by pressing and holding the Q. And here you will get a list of all the called quality scores. The quality scores and the bases are called by the sequencer itself. But if you wanted to get a summary of the quality scores for a specific window, you can choose Q over N option. This takes a window of 21 bases, 10 to the left and 10 to the right, and gives you a summary of the average quality in that area. So we'll turn this off and close this window. And we've set all of our parameters, and we've performed the vector scan. We'll go ahead and click on Assemble. And this assembly report again will show us what happened to all of our contigs and the default parameters that were selected. We can see here where each individual sequence went into which contig. And now we have a total of 4 contigs. And again, you can sort these by name, length, number of sequences. You can move them around depending on your preference. We'll look at the longest contig here, contig 2. And again, we'll go into the alignment view, double click on it. And we can scroll through and look at our aligned data to the consensus. Okay, one thing I wanted to show you is that in your alignment view, you can also look at your chromatograms. And to do that, you can click on one of these arrows. And if you wanted to view all chromatograms for all reads, you can hold down your Alt key and then click on the arrow, and that will expand all of the chromatograms for every single read in your assembly config. And here you can just get a quick snapshot of the average quality and the bases. And we can also click on the triangle next to consensus to look at the trace majority. So this is where I can show you where trace majority comes into play and our called base. So here you can see that the trace majority is S, and it chose that because of heterozygosity between the C's here, between the C and the G. However, if we zoom in, we can see that we have a G here and a G here, and there's some ambiguity. It's not clear, some overlapping peaks in this read. So we've gone ahead and taken the G's as the consensus base at this position versus a heterozygous call of S. So this would be more accurate than the trace majority, which was called. And you can go down and you can look at other examples. Here's another one. We have a trace majority of R because it took into consideration this A, which has some overlapping low-quality peaks. But it's quite obvious that G is the majority base at this position, so that's why we call this a G. And you can look at other examples in here as well. You can scroll down to look at additional reads. And then you can also view the quality scores by clicking on Q. If you press and hold the Q button down here, you can look at the sequencer's quality scores, or you can look at the average quality scores by clicking on Q over N. And this will give you a smoothened out version over a window of 21 for your bases. So I'll go ahead and switch them off. And I will shorten the chromatograms. One thing I wanted to mention is that if you're working with a larger data set, you might want to hide the sequence names. And to do that, you can simply click on this compact view button, and then that will hide your sequence names. Okay. And now I will open the strategy view. This is where you can look at your paired end information as well as your coverage report. And to do that, I will go into the config window and choose a strategy view. And here I can see the different coverages at the different areas. Typically, the higher the graph, the more coverage there is. We have the ruler, total sequence length of 2250 around. And then at the bottom, we have our reads. We have the forward and the reverse reads. We have the pair consistency. The more consistency there is between your pairs, the higher the graph there will be here as well. We can reorganize this. We can zoom in and zoom out. If you wanted to focus on a specific set of pairs, you can press and hold this button right here and choose this key. And this will allow you to focus on only pairs or non-pairs. And you can further choose some of these filtering options to focus on the specific types of reads that you might be interested in. Also, if you hover over any one of these reads, you'll get a summary of how big the distance is and where it's pointing to. So here, for example, we have a distance of, in this one, let's see, distance of 1,675. And it's pointing in the direction of contig 2, it looks like, the same contig, and here's the other read. And you can use, this information will be used in some of the gap closing steps for a genome, for example, or if you wanted to close the gaps between even a few contigs, like in this example. All of the gap closing options are built into the software in the config menu. You can see here we have the reassembling contigs, align contigs end-to-end. We can order the contigs from the project menu, and this will use a paired information as well. We recently did a webinar that focused specifically on gap closure for next generation sequencing data. However, all of the steps that were covered in that webinar will also be applicable to Sanger data. So if you're interested in gap closing, I would recommend that you view our webinar for gap closing that was done recently. So I will go ahead and close this window. And let's say you wanted to identify what these contigs are. You can again highlight a specific contig name and go into net search and blast selection. And you can choose the type of blast you wish to do and the database. And with this feature, you can also collect features from other hits that match your sequence. And to do that, you would simply highlight your specific hit, and you would click on Collect Features. And then it will annotate your sequence with that specific feature. And I will go ahead and close this window. It's taking a little bit longer than before. Actually, it looks like we just got some results here. So here we can see this is Enterobacter aeruginosus. And we'll go ahead and close this window now. You can also manually create features in your consensus. And to do that, you can simply go to an area of interest. You can highlight that region. And you go to Feature, New Consensus Feature. And here you can give it a name. And you can pick the specific type of feature this would be from the list. And we can choose miscellaneous feature for this. And now this will show up on our feature table. I'll go ahead and close this. I will switch back to the strategy view. I do want to highlight one point. If you wanted to improve areas, your coverage for your sequencing run, you can simply focus on that specific area, highlight it, or you can go directly into the contig menu and choose primer walk. And with this, if you're closing gaps, you can either walk into gaps. If you want to improve coverage, you can improve coverage, or you can do both. For our purpose, we'll just click on improve coverage, and you can then further define the parameters to meet your needs. Now again, if you're not sure what these parameters are, you can always click on the question mark, and that will take you to the help for that specific window. We'll stick with the default selection here and click on OK. And it will go to the list and give you a list of all of the primers that are found. So if you double click on any one of these primers, you'll be taken to that specific location in both the strategy view and the alignment view. I will shorten this a little bit so it's easier to see. And you can go down the list, and you can see the top and the bottom selections in both views. Again, this window, everything here is also sortable. You can click on score, sort by score, by index, primer sequence, or contig. If you had multiple contigs, you'll see a list of all the contigs here, and strandedness. And then you can look at additional information such as the length, GC content, delta G, distance, and so on. And all these can also be moved around. Okay. And when you're ready to order your contigs, you can always export this as well. You can go to File menu and choose Save Primer Info. And that is pretty much all I wanted to cover in our de novo assembly. And now I will move into our next workflow, in which we will cover assembling different multiple samples.
Farhan, before you move on to the next workflow, we have a question about editing. Is it possible to edit the consensus in SeqMan Pro?
Definitely. In order to edit, you would simply go to the location that you're interested in, and you can get there by using the conflict find or the Snip report or any other report, and you can just add gaps, or you can you can just add gaps or delete specific gaps in this example or bases.
Can you change the called bases in the reads or the sequence, the consensus?
You can change the consensus. You can add additional bases, for example, like a T here, but you cannot change the actual bases in the individual reads. These bases are called by the sequencer, and therefore they cannot be changed. But you can change, as I mentioned, the consensus, and then it will reflect, if you've created a conflict, it will reflect that in the report as a SNP.
Okay.
So it is possible to do that.
Great, thanks.
Okay, and with that, I will go ahead and move on to our next workflow. And again, to open a new project, I can go into File, New, and I'll go into Add Sequences. Okay, so now we'll start over. We'll highlight these reads and go into Assembling Groups. And then we specify our groups, and then now we're ready to assemble. And there we go. Now we have 3 contigs. Each contig has 2 sequences, and we can go into each one of them and open them up to see the forward reads and reverse. And a lot of times customers would like to perform a phylogenetic analysis on these. So in order to do that, you can simply highlight your contigs and send it directly to our Megaline program for the alignment. And to do that, you simply go into File menu and choose Send Consensus to Megaline. And here we have a list of all 3 contigs. And we can simply go into Align and choose one of the cluster alignment methods to perform the phylogenetic analysis. And we'll choose cluster W. And we'll quickly do the alignment. And now you can see the phylogenetic tree by going to View, Phylogenetic Tree. And here you can see our contigs that are very closely related. You can see a score, and we have a scale at the bottom of this window. And that was a very quick overview of assembling multiple sequences into groups. Many customers use a 96-well plate to do these types of sample assemblies, but you can also use it for other data types and experiments. And with that, I'll pass on the ball to Katie, and she'll take it from here. Thank you for joining.
Thanks, Farhan. We do have a few minutes for questions, so if there's any last-minute questions here, please feel free to chat those in. In the meantime, I'll sort of cover some administrative things. The first is that we do record these webinars, so if you missed any part of today's webinar, there'll be a recording on our webinars page probably within the next day or so, and we'll email everyone the link to that recording. So if you missed anything or want to watch any part again, there will be an opportunity to do that. Farhan also mentioned that we have some other webinars on things like gap closure. We also have one on Megaline and alignment. So if you want to see more in-depth analysis on some of the things Farhan touched on, we have extensive webinars on those with sort of these hour-long workflows. And the other resource we have is our video library, which is about 150 2 to 4 minute videos for the most part on one specific workflow. So we have videos covering some of the topics Farhan touched on today, as well as videos on other workflows using our software, including next-gen analysis, sequence alignment, Snip detection, etc. So that's a great resource as well if you want to learn more about the software.